Thank you. I think we have to find my slides on the computer first.
You need this? Yeah.
No, I have that right there. So while they're finding my slides, I just want to once again thank Esper for putting on just a fantastic course. And I hope all of the students here understand not the quality of most of the speakers, not myself included, but the rest of the speakers are really some of the best HIV and SIV researchers in the world, and it's you should be very proud to have all of these people come to Sao Paulo to teach this course. And they wouldn't do it were it not for Esper. So Esper, thank you. And I see my slides coming up now, so this is good. All right. Excellent. There. Perfect. Great, thank you very much. So yes, I am going to be talking about the impact of HIV and SIV on lymph node structure and function. What you heard about from Hendrik was mostly about aspects in peripheral blood. Let me see, I'm going to use that one. No, I'll try that one. Okay. So the lymph node is not just a bag of lymphocytes. It actually has secondary structures. This is from Janeway's textbook. Let me see. There we go. And what I'm going to be talking about are the T cell areas here in blue, the B cell areas in cream and yellow, and this structure called the germinal center. Now, this is a very simplified version of the lymph node. I'm going to make it even simpler. I'm going to take this one little structure here, and I'm going to turn it on its side and show it like this. So you have the lymphoid follicle, where the B cells are. You have the T cell areas in blue, and then you have the germinal center that is sort of at the junction of the B and T cells. And this is where the B and T cells come together to generate the antibody response. And this germinal center actually has its own structure. It's broken down into a light zone, which is where the T and B cells actually interact, and then a dark zone where the B cells then go to proliferate. The B cells that have rearranged B cell receptors that best recognize the antigen will actually survive, recycle back to the light zone, re-interact with T cells, and this process goes on several times, and that's how you get antibody maturation. This just shows what happens within that area. So once again, this is the B cell zone, and this is the germinal center. And the T cells, in order to get in there, these are the T follicular helper cells. What they do is, after interacting with dendritic cells and antigen at the border of the B cell zone, they upregulate CXCR5 and they downregulate CCR7. By downregulating CCR7, that allows them to move out of the T cell zone. By upregulating CXCR5, they can then follow the gradient of CXCL13 into the germinal center. In the process of doing this, they actually upregulate a master regulator called BC6. This keeps going in and out. And then they interact with germinal center B cells through a number of different molecules. They also liberate IL-21. The net result of all of that is that the B cell then upregulates activation-induced deaminase, which leads to somatic hypermutation and allows for affinity maturation and changes in the B cell receptor, leading to changes in the affinity of the immunoglobulins that these B cells are producing, and that's how you get affinity maturation. What I will talk about in this lecture is first background on T follicular helper cells, which I've mostly given you, and then talk about SIV infection, SIV88 infection, and association with broadly neutralizing antibodies. And then I'll go on and I'll talk about CD8 T cells in lymph nodes. So first, T follicular helper cells. So as you heard, and as you all know, with HIV infection, you get loss of CD4 T cells. But in the lymph node, what I'm going to tell you is that most of that loss is actually loss of CD4 T cells from the T cell zone. And in fact, what you get is accumulation of CD4 T cells in germinal centers. And that was shown, and you saw the data in Hendrik's talk, from two papers that were published at the same time, one from my group looking at SIV-infected lymph nodes, and from Hendrik's group looking at HIV-infected lymph nodes. And the data look like this, that with SIV infection on the left, or HIV infection on the right, instead of seeing a decrease in T follicular helper cells, you actually see in about half of the monkeys an increase in T follicular helper cells. And in humans, you see a number of individuals have this increase in T follicular helper cells. This is what it looks like when you actually look at a lymph node. So this is an SIV uninfected lymph node. This is a germinal center. I blow that up so you can see it here. This is the light zone, where you have CD4 cells that co-express PD-1. You can see them up here. And then here's the dark zone. So these are the B cells which are expressing Ki67, a proliferation marker. So these are the B cells that are proliferating. So this is a standard germinal center in an SIV uninfected lymph node. In SIV infection, however, what you see are these much larger germinal centers where you have this very large light zone where you have a lot of T follicular helper cells interacting with B cells, and this area of B cell proliferation. And if you look, you can see you have a number of these big expanded, disorganized germinal centers. This is the lymphadenopathy that was pathognomonic for HIV infection back when those older of us in the room used to see individuals with immune deficiency. We would palpate lymph nodes, we would feel this, and we would make the diagnosis of HIV infection before we had diagnostic tests. Now, what is driving this accumulation of T follicular helper cells? I'm not going to go through all the data that we published in the JCI paper, but I'll just summarize it here. So we looked at a number of different things. We first asked, is it infection of the different T cell subsets? And we found that no, that wasn't it. Whether you looked at the T cells outside the germinal center or inside the germinal center, they were all equally infected. So it wasn't that these cells out here were getting infected and dying, and these were being protected, and so they were surviving. We asked whether it was an effect of T cell turnover. So we simply asked, what's the rate of proliferation of these cells versus these? And we did not see that these cells were turning over faster. So it wasn't that once the cells got in here, they were proliferating and expanding. In addition, we looked at their susceptibility to death, and what we found is that these cells in here were actually more susceptible to cell death. So it wasn't that once they got in there, they weren't dying, and that's why they were accumulating. So the only thing we could come up with was that SIV infection was somehow driving a drive towards differentiation of T follicular helper cells. And that's actually what we found was going on. And what was driving that was immune activation. What we found is that with immune activation, you got increased IL-6. That was altering STAT phosphorylation patterns within these T cells, leading to an increased drive to T follicular helper cell differentiation and away from Th1 differentiation. So it was actually SIV and immune activation that was leading to this drive. And this will be sort of a theme throughout my talk, and in subsequent days, you'll hear a lot about immune activation. But immune activation is actually driving a lot of what's going on in these lymph nodes. So with these increased germinal centers, what's happening to the B cells? When we looked, we said, let's look for antigen-specific B cells within the germinal centers. The way you can look at that is for B cells that bind peanut agglutinin. As shown here, you only find those cells in lymph nodes. You don't find them in peripheral blood. And then what you can do is simply ask within lymph nodes of SIV-infected monkeys that have high T follicular helper cells, what you see is that you have a lot more cells that bind peanut agglutinin, so more germinal center B cells. They express higher levels of BCL6, and in the peripheral blood, you find higher levels of antibody. And this just shows GP120 antibody, but you can also see the same for for GAG-specific antibody. So this process is driving more antibody production. So if you're getting more antibody production, does that mean you're actually getting better neutralizing antibody production? So we decided that we could actually look at this if we looked not at SIV-infected monkeys, but at SHIV-infected monkeys. And the model we looked at was one that was published by Mal Martin and Dennis Burton several years ago, SHIV88. What they discovered in this paper in PNAS is that a number of these monkeys who were infected with SHIV88 and were chronically infected actually developed broadly neutralizing antibodies. So what we asked was, can SHIV88 infection in macaques be used to study factors associated with the generation of broadly neutralizing antibodies, looking specifically at T follicular helper cells? And this was work that was published a couple of years ago by Takuya Yamamoto, who was a postdoctoral fellow in my lab at the time, came from Tetsuro's lab, and is now at Osaka University. And Rebecca Lynch, who's sitting in the second row there, and I think you haven't spoken yet. You'll be hearing from her later in the course. So what we did is we took 8 monkeys, infected them with SHIV88, and then took multiple lymph node biopsies over time and blood and evaluated what was going on. What we knew from past studies with this infection is that neutralizing antibodies come up slowly, but by about 44 weeks, you can usually find broadly neutralizing antibodies in these monkeys. So we tested the serum at week 44 against a number of different viruses shown here, and if they had high titers, those are shown in red, medium titers in yellow, low titers in green, or absent, and we could basically total up all of the neutralizing titers against all of these different viruses and come up with a neutralization score. The higher the number, the more broad and potent the neutralization is. What you can see is three of the monkeys are shown in red and will be throughout the rest of the talk as having the strongest and broadest neutralization. The ones in blue have lower neutralization, and the ones in black have weak neutralization. So the first thing we asked is what was associated with broad neutralization. And the first thing we looked at is just viral load and CD4. And interestingly, the monkeys that developed the best neutralizing antibodies had the highest viral loads and the lowest CD4 count. So these were the sickest monkeys, but they had the best neutralizing antibody response. So then we asked, well, what about T follicular helper cells? By measuring T follicular helper cells over time in these monkeys in their lymph nodes, what we saw is that you see the red and the blue monkeys tend to have more T follicular helper cells, but it's not as good a correlation as we saw over here. So we decided to look at antigen-specific B cells and antigen-specific T follicular helper cells. First, the B cells. So envelope-specific B cells in the peripheral blood, shown on this axis, versus the neutralization score on this axis. Peripheral blood B cells don't predict neutralization at all. But if you look in the lymph node, the germinal center envelope-specific B cells are an excellent predictor of whether that monkey is going to have a broad neutralizing antibody response. What about T follicular helper cells? So, as I mentioned, one of the best cytokines to look at is IL-21. Unfortunately, it's very difficult to measure IL-21 after stimulation. So what we did is we looked at mobilization of CD154 or CD40 ligand. And what you see is a reasonable correlation after stimulation with envelope peptides between envelope-specific T follicular helper cells by CD154 and neutralization score. That correlation gets much better if you look at a T follicular helper cell type of cytokine such as IL-4. So here you have a better correlation. However, if you looked at a Th1 cytokine like interferon gamma, the correlation was terrible. Now remember, all of these cells over here are phenotypically T follicular helper cells. So they're in the germinal center, but they're not making IL-4 after stimulation. They're making a lot of interferon gamma. And what that means is that even though a cell is phenotypically a T follicular helper cell, it doesn't necessarily make the right cytokines. So there's a quality to T follicular helper cells. So to look at that more closely, we sorted envelope-specific T follicular helper cells from monkey lymph nodes based upon just mobilization of CD154, isolated RNA, and performed qC-PCR for 11 transcripts associated with lymph node function. And here's the way the data broke out. So what you see, once again, is the monkeys that have good neutralizing antibodies. If you look at their T follicular helper cells, they make a lot of BCL6, CXCL13, MAF, IL-21, but don't make these Th1 type of cytokines. The bad neutralizing monkeys do the opposite. Their T follicular helper cells don't make things that you would expect of T follicular helper cells. They make things you think of for Th1. So, they have bad T follicular helper cells. T follicular helper cells that are more like Th1 cells. The other thing that we wanted to look at is, in order to get good neutralization, you usually need a lot of somatic hypermutation. So what does the somatic hypermutation look like in the monkeys with good neutralization? Well, we sorted their B cells, we did sequencing, we looked at how much somatic hypermutation their SIV envelope or HIV envelope specific B cells had, and what you can see is the better the neutralization score, the higher their amount of somatic hypermutation. So the monkeys that were developing good neutralization had good T follicular helper cells and lots of somatic hypermutation. And finally, in humans, it had been shown that the envelope sequence diversity had to drive a lot of this. So we simply did single genome analysis of envelopes, the HIV envelopes from these SIV-infected monkeys. These show the two monkeys who had bad neutralization. Their envelopes really weren't evolving. Two monkeys that had intermediate neutralization, their envelopes were evolving over time, but the three monkeys that had great neutralization had lots of envelope sequence diversity over time. So, what does all this mean? It means that SIV88 infection can actually be used to study factors associated with the generation of broadly neutralizing antibodies. They develop slowly over time and are associated with high viral loads, low CD4 T cell counts, more and better quality of envelope-specific T follicular helper cells in lymph nodes, more envelope-specific germinal center B cells, greater somatic hypermutation, and evolving Env sequence diversity. What this probably means for vaccine development is that antigenic changes will be required for Env immunogens to drive the degree of somatic hypermutation required for broad neutralization, but we can use these types of non-human primate studies to study that. I have to tell you that about 2 years ago, I presented these data here, and Ron DeRozier was in the audience, and he heard the data, and we planned an experiment to actually test this theory. And I think next week, we're actually going to get started on that experiment. So it's been a long time coming, but we're going to get there. All right. Let me move on for the second half of the talk and tell you about CD8 T cells in lymph nodes. Why CD8 T cells in lymph nodes? Well, you've heard that T follicular helper cells are infected. There's lots of virus replication there. They're important. And CD8 T cells, as you know, are one of the major ways that virus is controlled in humans and in monkeys. But what the problem appears to be is that from a number of studies from Liz Connick's group, where you can see CD8 T cells in lymph nodes and in germinal centers, there really aren't a lot of HIV or SIV specific CD8 T cells that get into the germinal center, where most of the virus is replicating. So what we wanted to do is perform a comprehensive analysis of the phenotype, location, and cytolytic killing activity of lymph node CD8 T cells during both HIV and SIV infection, and then study the role of viral replication and other factors to figure out what's causing the trafficking or what's inhibiting the trafficking of CD8 T cells into germinal centers. So some of the data I'm going to present to you were just published 2 months ago by Kostas Petrovis and Sarah Ferrando Martinez in my lab, but I'll tell you more unpublished data. What was published 2 months ago was on HIV infected in human lymph nodes. I'll tell you a lot more about what we've been doing with SIV infected lymph nodes and more of the trafficking story. So what we found is that when we looked for CD8 T cells in lymph nodes, so this is HIV-infected lymph node, what you see is that in an HIV-uninfected lymph node or an HIV-infected lymph node, what you see is in HIV-infected lymph nodes, you get this population coming up of CD8 T cells that are CCR7 negative and CXCR5 positive. And so these are cells that should be moving into germinal centers because they've lost CCR7 so they can get out of the T cell zone, and they express CXCR5 so they can follow a gradient of CXCL13. If you look at total CD8 T cells, either in monkeys or in humans, what you see is that with SIV infection, you get an increase in total CD8 T cells, at least as a percentage of T cells, and compared to either tonsils in T or HIV-uninfected lymph nodes, that in HIV-infected lymph nodes, whether you are or are not on antiretroviral therapy, and the yellow are people who have been on antiretroviral therapy for a long time, you once again see this increase in CD8 T cells. Well, this is total CD8 T cells. What about these, what we call follicular CD8 T cells that express high levels of CXCR5 and don't express CCR7? What you see is, once again, within SIV-infected monkeys, you have an increase in these follicular CD8 T cells, and the same thing in HIV-infected humans, an increase in follicular CD8 T cells. Now, I call these follicular CD8 T cells just based upon their phenotype, but are they actually in the germinal centers? Well, we can use a technique called histocytometry, where we identify actual germinal centers based upon co-expression of B cell markers like CD20 and Ki67. So we can identify germinal center 1, germinal center 2, germinal center 3, and then we can go in and actually count the number of CD4 and CD8 T cells within each germinal center. And we can combine those data and actually ask not what's the percentage, but the actual number of CD4 and CD8 T cells in the germinal centers. When you do that in SIV infection, what you see is in chronic SIV infection, you get an increase in the total number of CD8 T cells in germinal centers. And the same thing in HIV infection. With HIV infection, you get an increase in the total number of CD8 T cells in those germinal centers. So these are CD8 T cells that are actually moving into the germinal centers. This just shows those same data. It may be hard to see here, but what we have is a germinal center in a tonsil, an HIV-uninfected lymph node, an HIV-infected lymph node, and an HIV-infected lymph node on antiretroviral therapy. You can see CD4 T cells in all of these. What you probably can't see is that there are virtually no CD8 T cells here or here, but you can see some red cells here. These are the CD8 T cells, and here there are CD8 T cells throughout. So you can actually see the CD8 T cells in the germinal center. You can do the same thing with SIV-infected monkeys. It becomes a little more difficult because we don't have a CD8 antibody that binds well for this type of analysis. So what you have to do is you have to look for CD3 positive cells that are CD4 negative. The CD8 antibody works perfectly fine in flow cytometry, but not in confocal for some reason. So anyway, what you can see here in HIV or SIV-infected lymph nodes are these germinal center structures, and if you look, there are red cells that don't express green. So these are CD8 T cells, and the reason we can do that is we know that there's a very, very small population of cells that are double negative, so that don't express CD4 or CD8. So we can pretty much say that if you express CD3, but you don't express CD4, you're CD8. Now, the other thing I showed you is that the lymph node and the germinal center structure starts to break down with SIV infection and HIV infection. So what you can get are these B cell follicles, which are pretty intact, or you can get them that really just start to break down. And so it has been suggested that it's only when you get complete breakdown of your germinal center and your B cell follicle structure that the CD8 T cells can get in. So we looked at lymph nodes that either had a fairly intact follicular structure or very disorganized follicular structure. And once again, identified the germinal centers and counted CD8 T cells. And what you can see, here's looking at the actual number of CD8 T cells in these germinal centers. Whoops, I hit the wrong button. What you can see is that with chronic SIV infection, with an intact follicular structure, you do start to see an increase in the number of CD8 T cells. But where you really get the big increase is when that follicular structure starts to break down. So I think the CD8 T cells can start to move in even when the follicle is intact. But once it starts to break down, then you really get movement of those cells. So are those CD8 T cells that move in, are they HIV or SIV specific? So we can stimulate with peptides, and we can look for cytokine production. We can ask, is there enrichment of SIV or HIV specific CD8 T cells in the follicular CD8s versus the non-follicular CD8s? And this shows in non-human primates with gag or envelope stimulation, the non-follicular CD8s and the follicular CD8s look exactly the same. Exactly the same thing if you look in humans in the lymph nodes. So there's no specific enrichment in the germinal centers of HIV or SIV specific CD8 T cells. Are these cells, do they, what I won't show you, but what was in the paper is that they are somewhat defective in cytokine production, but they appear to have good cytolytic potential. So this shows their granzyme and perforin content of follicular CD8 T cells in an HIV-infected lymph node. Those follicular CD8 T cells have a lot of co-expression of granzyme and perforin. Once you put them on antiretroviral therapy, that goes down, however. If you stimulate those cells in vitro and ask about their expression of granzyme or perforin, what you see is that granzyme goes up in the follicular CD8 T cells in HIV-infected lymph nodes quite nicely. Perforin also goes up, but it appears that perforin, with the introduction of antiretroviral therapy, the perforin, once again, goes back down. Granzyme doesn't. Granzyme stays up after stimulation. And the same thing appears to be true in SIV-infected lymph nodes. One of the ways that we measure these, it's really the effector follicular CD8 T cells that we need to look at. And what you can see is that they, in SIV-infected lymph node samples, they have nice expression of granzyme and perforin. Another way to look at this is once again by confocal microscopy. So here's an HIV uninfected lymph node, HIV infected, and an HIV infected lymph node on antiretroviral therapy, looking at CD8 and granzyme. And what you can see here is that in the HIV infected lymph node, whether you are on antiretroviral therapy or not, you have a lot of CD8 cells in red that co-express granzyme. Once again, in the SIV-infected monkey lymph node, what you have to look at, and I'll blow it up here, this is a CD3 positive cell that expresses CD4. It does not express granzyme in green. This is a CD3 cell because it expresses red. It does not express CD4, the purple. But it expresses granzyme. So the CD8 T cells do express granzyme. How can we actually measure effectively whether this cytolytic potential, whether these cells can actually kill? Well, the way we do this is to use a bispecific antibody. So in this case, it's a bispecific antibody, half of which is VRC07, which recognizes HIV envelope. The other half is an anti-CD3. These can be produced quite nicely. And what an antibody or a bispecific antibody like this does is the anti-CD3 portion will bind to CD3 on any CD8 T cell and activate that T cell. And then the other half will recognize envelope on the surface of an HIV-expressing cell, and this will bring these two cells together. You'll get redirected killing, and you'll be able to say that this cell has real cytolytic potential. So when we do this type of assay with different populations sorted from lymph nodes, what we find is that the population from either a tonsil or an HIV-infected lymph node that has the best cytolytic capacity is the CCR7 low, CXCR5 high cell. And that's true in an HIV-infected lymph node or an SIV-infected monkey lymph node. So these follicular CD8 T cells with cytolytic potential are in the germinal center. What's bringing them in there? What's the driving force behind this? A lot of people like to think that, well, there's HIV replication and those CD8 T cells are following that antigen gradient. But actually, T cell receptors don't follow antigen gradients. Other things drive CD8 T cells to move. So what we did is we looked within HIV-infected lymph nodes, within their T follicular helper cells, in 4 lymph nodes from HIV viremic individuals, 4 individuals who were on antiretroviral therapy, and we looked at the level of HIV RNA, either GAG, TAT, or REV. And you see these viremic individuals, you could detect lots of viral RNA being expressed in their T follicular helper cells. In those that were on antiretroviral therapy, you couldn't. We then looked at their percentage of follicular CD8 T cells along this axis versus the RNA. Sorry, my thumbs are too big. What you see is all these individuals down here are the ones who were on antiretroviral therapy. All right, so no viral antigen that you could detect, yet they have this range of follicular CD8 T cells. These monkeys, or humans, had a range of virus and somewhat of a range of follicular CD8 T cells, and there's no correlation whatsoever. So it's not viral antigen that's driving this. The other way we can look at this is by using RNA-scope in SIV-infected lymph nodes. So here's an SIV-uninfected lymph node. Here are 2 SIV-infected lymph nodes. You can use SIV RNA-scope to look and find the SIV RNA in these lymph nodes and actually identify where it is. And you actually see a couple of different patterns. So you can find a pattern like this where you see a cell where the whole cytoplasm is just full of SIV RNA. So that's almost certainly a productively infected cell. But you also can find patterns like this where you just get these little dots of SIV RNA. And this is almost certainly RNA that's floating around and maybe binding to the surface of the cell or is on the follicular dendritic cell network. So here I show you monkeys with 4 different levels of virus from lowest to highest. Here you can see the germinal centers. We can go in and we can identify follicular CD8 T cells, and here we can identify SIV RNA, and because of the lights you really can't see this, but what I can tell you is that there's no correlation between SIV RNA and the number of follicular CD8 T cells that we see. So if it's not SIV or HIV expression that's driving these cells in there, what could it be? Well, immune activation and inflammatory signals, as I told you, are something that seems to drive a lot of what's going on. Now, what we know is that SIV infection of Rhesus macaques leads to lots of immune activation and pathogenesis. SIV infection of African green monkeys, you get a lot of viral replication, but you don't get inflammation. So if we looked at African green monkey lymph nodes, which don't have immune activation, do we see follicular CD8 T cells? And the answer is no, we do not. So in African green monkeys, we don't see this accumulation of follicular CD8 T cells in SIV-infected African green monkeys. In SIV-infected Rhesus monkeys, we do. So inflammation here, accumulation, no inflammation, we don't see the accumulation. So what are the inflammatory markers that might be going on? Well, we know that in SIV-infected monkeys, you get this in lymph nodes, you get this accumulation of activated monocytes, CD16 and CD14. This population is much bigger than this population. And in SIV-infected monkey lymph nodes, you see this increase in CD14, CD16 positive monocytes. You don't see it in SIV uninfected monkeys or in African green monkeys. There's a nice correlation then between the number of these monocytes and the number of lymph node CD8 T cells. This can be seen visually here. So this is an SIV uninfected monkey. This is an acutely SIV infected monkey, and this is chronically SIV infected monkey. What you have here is in the red and the green is CD163, a monocyte marker, and myeloperoxidase. So these are activated monocytes, and what you see is this incredible accumulation of these activated monocytes with SIV infection. You see the same thing in HIV infection. So here's an HIV uninfected lymph node, CD163 and myeloperoxidase. You don't see many of these monocytes. With HIV infection or HIV infection on antiretroviral therapy, these cells are all over the place. So what do these activated monocytes make? One of the things that they tend to make is CXCL10 or IP10. That is a ligand for CXCR3, and CXCR3 expressing T cells will follow a CXCL10 gradient. We know that from either interferon gamma or interferon alpha expression, you will get monocytes to express CXCL10. So what we asked is, after interferon alpha stimulation or interferon gamma stimulation, could we see CXCL10 expression in either CD14, CD16 positive, double positive monocytes, or just CD14 positive monocytes from SIV uninfected or infected monkeys. And what you see is that with SIV infection, you get increased CXCL10 expression after interferon alpha stimulation. No real difference with interferon gamma stimulation, though they do express CXCL10. What about CXCR3 expression? If the CD8 T cells are going to follow that gradient of CXCL10, they better express CXCR3. Well, in SIV-infected lymph nodes, virtually all of the CD8 T cells express CXCR3. That's not true in the peripheral blood. About half of the cells, CD8 T cells in the peripheral blood express CXCR3, but once you're in the lymph node, they almost all express CXCR3. And this shows, so this is in humans, and this, I'm sorry, this is in SIV-infected peripheral blood and lymph node, and this is within the actual follicular CD8 T cells. This is total, and this is follicular CD8 T cells. And finally, do those cells that express CXCR3 and would be responding, do they express granzyme, so are they cytolytic? And the answer is yes. In the lymph node, virtually all of these CXCR3 positive follicular CD8 T cells express granzyme. So what does this, one last bit of data. So I've said they should be able to follow this gradient of CXCL10. Do we have any evidence that they actually can migrate in response to CXCL10? Well, one assay that you can do to look at this is called the phalloidin assay. You take cells, you label them with phalloidin. If they start to respond by migrating, they have to start to polymerize actin. What phalloidin does is measure that polymerization of actin. So what you have here is an SIV uninfected lymph node, and this should be SIV infected. We can look at memory CD3 cells. We can look at CD8 cells CD8 cells that are naive or memory in the presence of CXCL10. And what you see is in SIV-infected monkeys, their memory CD8 T cells actually start to polymerize actin in response to CXCL10. That doesn't happen with the naive cells. It's only the memory CD8 T cells. And that's shown better here. So this is a measure, as I say, of chemotaxis. No CXCL10. Your polymerization is down here. With CXCL10, you start to polymerize actin. So a nice measure of chemotaxis. So what does this all mean? Well, when you have HIV infection up here in your B cell follicle, what you probably do is you start to create activation. What that activation does is lead to the bringing in of monocytes that express CXCL10. These follicular CD8 T cells start to follow that gradient and end up moving up into the germinal center because of their expression of CXCR3. Now, once they get there, they're not necessarily SIV or HIV specific, so they can't really kill these cells. But if we now go in with a bispecific antibody, we can now make these cytolytic CD8 T cells kill those cells and start to eliminate the virally infected cells within those germinal centers. So, CD8 T cells do accumulate within the B cell follicle and the germinal center during HIV and SIV infection. Follicular CD8 T cells are functional. They're capable of mediating bispecific antibody-directed killing of both HIV and SIV-infected cells. And immune activation seems to be the major force driving the recruitment of non-naive, so memory CD8 T cells into the lymph nodes and germinal centers. And the use of bispecific antibodies may be able to take these cells that aren't HIV or SIV specific and get them to actually target the HIV infection in these germinal centers. I think I've mentioned during this talk of the individuals who are involved in this work, so I'll just leave this slide up here. Thank you for your time, and I'm happy to take any questions you might have.